morning, everyone. Today we're going to talk about nitrogen cycling in cover crops and how the nitrogen can or cannot move to the next crop. So this is a very simplified nitrogen cycle showing here we have cover crops that we can plant after wheat or another crop in the fall. We have brassicas, grasses, and we have legumes that fix nitrogen from the atmosphere and move it right here on the nodules or fixed symbiotic fixation, and then nitrogen goes into the crop and into the soil. And then the decomposition of these crops, before, we have residual nitrogen that is taken up by these crops. And even legumes that fix nitrogen, they also take quite a bit of nitrogen up. Now, when these crops get winter killed, or they go back to the soil as residues, and as you see here, is very large tissue, has low and carbon nitrogen ratio. And because of that, the books and all textbooks say that low CN ratio will lead to a release of the nitrogen as ammonia into the soil for the next crop. And there's also going to be mineralization of the organic matter in the spring when we plant this crop. And that ammonia is going to nitrify and form nitrate, which is taken up by the cash crop, in this case corn, and also plants can also take ammonia directly. Then we have nitrogen from fertilizer, which in our areas mainly comes out of ammonia form, which is also nitrified and goes to nitrate. And we have several other losses from the system as volatilization of ammonia, denitrification, and leaching. And we can have some immobilization of the ammonium to organic matter. So this is what the cycle will be. And so we will expect that the nitrogen in these plants, whether it was taken up from the soil or fixed, should be released for the plant's use. So let's see what we find out when we try to answer this question. First, let's see how much nitrogen actually the cover crops take up. And here you can see we have 3 legumes, forage pea, Austrian winter pea, hairy vetch, and 3 brassicas, forage turnip, purple top turnip, and forage radish. And here you can see the nitrogen accumulation in pounds per acre and the biomass of these crops that were planted on August 15 and harvested on November 15. So about 4 months of growth. And the nitrogen in the biomass was the highest for forage pea with 120. And you see all the legumes have more than the brassicas because the brassicas, most of the nitrogen comes from scavenging from their residues, while in the legumes, the nitrogen comes from both scavenging, legumes are good scavengers, and nitrogen fixation. Legumes usually use nitrogen from the soil before they start fixing nitrogen. Another important thing on how much nitrogen is actually in the biomass of cover crops is the seeding date, which actually drives growth. And it depends also on the crops, what species we have and when we plant them. In this graph, we have many of them, many different cover crops, annual grasses, brassicas, legumes, and other families, planted on 2 dates, 29th of July and 3 weeks later, and August 23rd. And the blue bars indicate the first date, and here we see the biomass that these crops produce. So in biomass terms, you can see that forage sorghum, for example, which is a warm season, in the first date had over 1 ton per acre yield, and then the second date it dropped completely, very low because it froze very soon after planted. So warm seasons don't have much time to grow and don't have much time to accumulate. But even cool seasons can be affected by the planting date because there's less time to grow and accumulate nitrogen. So this is directly related to nitrogen. And here you can see some pictures where forage sorghum, we have date 1, late July, day 2, 3 weeks later. You see there's almost no biomass. There's no effect on cover. There's no cover on really the soil, but it certainly is not taking up any nitrogen. Same with millet, another group cool warm season. In cool seasons here, you can see there is not so much effect, although you can have some because less time growing in the soil. Now, brassicas also will be affected in growth, not as much as warm seasons, but there is less time for these crops, especially these ones, they're scavengers like turnips and radish from taking up nitrogen from the soil that we are hoping it protects it from bleaching and then it's moving to the next cash crop. Another thing is important is how much soil residual nitrate actually it is available in the soil when you plant the cover crop that is not a legume. Here you can see radishes and turnips planted in two different sites, same year at same time. And one site had only 30 pounds of nitrogen between 0 and 24 inches in the soil of residual nitrogen, while in the other location we have over 100 pounds of nitrogen per acre and the same depth. Both had about the same rain, planted at the same time, harvested about the same time. You can see the huge differences in sizes. So if the crop does not have nitrogen to grow, even if it has all the other conditions, it's going to stay very small. And a small plant takes up very little nitrogen compared to these plants here that you can see there's a lot of tissue, very lush. And actually you can see that if we sum the root and the leaf tissue, we have more than actually we measure in the soil, and that's because these roots probably went deeper than 24 inches. Now, another thing is important, most of this nitrogen is in the leaves and not on the roots of the plants. So we've done a lot of research that we've measured how much nitrogen we have in the biomass of cover crops in the fall. But about 3 years ago, we start thinking, okay, we need to know now, what is this nitrogen going? Is it going to the corn? Is it going to the sugar beets? So I'll show you some of the data from this research of where we evaluated several cover crops and see if the nitrogen that we see in the fall actually it is being taken up by the corn. And here for this experiment, we have fava bean and forage pea, which are legumes, and winter camelina, which is a brassica, and it's a winter hardy brassica. We did not have grasses on this study because we already know that rye can have some toxicity problems or nitrogen immobilization before corn. So we want to see if the nitrogen in the legumes was moving to the corn. So first, how much nitrogen do the cover crops take up actually? And here you can see we have different locations and years, okay, 2 years, 2 locations. This is in the fall, planted right after wheat, and then harvested sometime at the end of October, November. And you can see that the nitrogen taken up by these plants depends a lot on season. Okay, in season because of if we have water for the cover crop to grow, if we have how long is the season for it to grow. You can see in 2017 at this location, we have all the conditions for the cover crop to grow, and they grow really fast. And fava bean had the most. Both fava bean and forage beet had the most growth and the most nitrogen. Camelina is a winter crop, so we don't expect it to produce as much biomass in the fall. You see it vary even in some years. Camelina, it was there, but it was so small we couldn't actually measure nitrogen in it. And you can see vary with the different years what crop did the best. But I want you to remember this number. Here we have 140 pounds of nitrogen in the biomass of fava bean, and this is what we want to see, can we claim any nutrient credit to the corn from this nitrogen here. So that's about 3,000 pounds of per acre of biomass. That's how much biomass this crop had. So clearly the growth and the nitrogen that the cover crop takes up will depend on the season, on the crop species. If the legume is going to be higher, and you saw in the other data too, because part is from scavenging, but part is from nitrogen fixation compared to the non-legume. And definitely crop growth is going to drive that nitrogen uptake. So what happened? In the following year, where we have these cover crops plots, we planted corn, but also we had different nitrogen rates to be able to test if some of the nitrogen from the cover crop was cycling to the corn. So we had 0, 40, 80, 120, and 160 pounds of nitrogen per acre. And then each one of these lines is one of the cover crops. So what we expect, if there's any cycling from the cover crop to the corn, we will expect that at 0, we will have a higher yield on the cover crops than the check. Check line are these black triangles, and you can see that winter camelina actually had lower yield at all nitrogen rates. Now, at all nitrogen rates, corn responded, increasing yield, regardless what cover crop we had. That indicates you that all those 140 pounds that were on the fava bean did not go into the corn. And so we had some significant differences from the check and 4-HP. So some indication that it was a little bit higher than the check, not much though. Some indication that maybe some of the nitrate did go into the corn, but nearly enough to call it a nitrogen credit that we could claim for that. So this study done over 2 seasons and 2 locations, we could not really claim that we have any nitrogen credit for any of these cover crops and corn. One important thing is to consider, especially when you're using winter hardy cover crops like camelina, is that there is an interaction with gravimetric water content, soil water content. The spring of 2018 was very dry, and since camelina grew in the spring, it took more water significantly than all the other treatments that were winter kill. So definitely this is something we need to consider, because here's in a confounding effect that the winter crop, even if it could have cycled some nitrogen, is taking too much water in the spring to be damaging the corn. It will cause an effect on establishment and even in yield. So it's very important to consider that. Let's move to sugar beet. We did a very similar study in sugar beet where we have more cover crops. We have 6 different treatments of cover crops and only 2 nitrogen rates to try to test the same thing. Can we see any of the nitrogen from the cover crops into the sugar beet the following year? So here the cover crops were 3 winter crops: winter rye, winter wheat, and winter camelina. And then oats and radish we will winter kill, and then the check plot with no cover crop. We did not use legumes here because sugar beet quality, I mean content of sucrose in the root, can decrease if there is late nitrogen in the season. So a late release of nitrogen from the legume could affect that. So we did not consider a legume in the trial. So what happened? First, again, how much the nitrogen the cover crops took up. You know, and in the fall, all of them were similar, except for radish, which had a higher nitrogen uptake than the other crops. And this, we can expect it. Radish has a longer root and can draw more, can scavenge more nitrogen from the depth. Now, 3 of the cover crops we have did survive the winter and came back in the spring, and they had nitrogen, they took up nitrogen again. It's important to know whatever leaves or biomass that took up nitrogen is going to be dead in the winter. It's burned up, all the leaves. All is left really are the buds, and then so the nitrogen uptake we measure in the spring is new nitrogen taken out. So we sum both of them now. Obviously, all winter crops are much higher than the oats and radish, which winter kill. So we had up to, if we consider both seasons where the crop grew and took up nitrogen, we can see that we had up to 100 pounds. So again, you see every time we measure this, we're kind of seeing a similar trend, right? This is for cover crops. And this is good because that means these crops are taking residual nitrogen and the soil there actually could be lost to the system by leaching. And again, yield-dependent crop species, if it winter kills or no winter hardy or winter hardy, right? There's a huge difference in how much it's going to take. And again, crop growth is going to drive the nitrogen uptake, so it'll depend on the season and the water used in the spring. If there is enough water for the crop, it will grow more and take more nitrogen. So what happened? We did not see any nitrogen fertilizer effect, so I'm not going to show it. We had only two rates, 0 and 100, and there wasn't any difference between cover crops or nitrogen. That means we did not have any effect of nitrogen in sugar beet. Now, you can see again that the seasons make a huge difference in yield. Okay, and we have some differences here. One difference was in the treatment they had winter rye from the season before. So here, winter rye affected negatively the yield of sugar beet. And this, again, like what happened with winter camelina and corn, this was because of water use, mainly. So in recoverable sugar, we also saw a problem in here. It was not only rye, but it was only in camelina. So two of the winter hardy crops reduce recoverable sugar in two of the locations in years. So we're having a negative effect of these winter hardy cover crops. No effect on any of the other ones, and like I said, no effect of nitrogen, so no nutrient cycling. And again, the interaction was happening with water. Both winter camelina and winter rye use more water in the spring than any of the other treatments. They have been winter kill or they didn't have a cover crop. This difference, although it might seem small, was very important. And here in this picture, you can see this is a picture taken with a drone, the real picture and then the NDVI picture, and you can see that the three treatments with the winter crop, rye, camelina, and wheat, had much less establishment. Now, this doesn't mean there weren't any plants. Sugar beet plants were smaller, so they weren't detected by the NDVI. But these carry on the whole season, and that's why those treatments end up having winter rye and winter camelina end up having a lower yield at the end of the season. Both crops were terminated right before we planted sugar beet, but even these pictures are after we applied glyphosate, plants don't die right away. So they were having some competition and they were using water in a very dry season. So the question we wanted to answer is, do we have nutrient credits from cover crops to corn or sugar beet? Is no, we couldn't see it. And so the question, we know that cover crops scavenge nitrogen from the soil. We saw it every time. And that's a good thing, right? Taking that nitrogen, that nitrate from the soil so it cannot leach. We can see that it depends on the seeding day, water availability, soil residual nitrogen, cover crop species, right? If it's legume, brassica, if it's a winter hardy or not. But also so that we have to watch with water if we're using winter hardy cover crops. Now the point in what we wanted to answer, right, we couldn't, and nitrogen in cover crops biomass did not cycle to the next crop. Why is that? We really don't know. If you remember the cycle I presented at the beginning, what are we missing on this cycle? This is a typical cycle from any textbook that says that if we have these crops, that nitrogen should be released and plants should be able to use it. But that's not happening, so what are we missing? And Dr. David Franzen from the Soils Department at NDSU, he's working on several theories, and one of those is the presence of this, we have soils with clays called smectites. They're high shrink soil. That means when it's wet, the layers on this clay open up, and then when it dries, they shrink. When that happens, they can trap ions, and ammonia is an ion. And because of that, we could be some of that ammonia might be being fixed, temporarily fixed while the soil is dry, and in those dry springs, it just wasn't able to release, because as soon as it came out of these crops, it went into the clays instead of going into the soil solution to be available for the corn. We don't know yet, but the first soil test of non-extractable ammonia that Dr. Franzen did show that the plots that they had cover crops had much higher non-extractable ammonia. So it could be one of the explanations of what is happening. But unfortunately, if any farmer asks, can I consider any nitrogen credit of cover crops to corn or sugar beet? And we have to say no. We have not been able to see that. And this has been the result of many of the studies we've done in different in our environments. With that, thanks so much and for your attention and interest. Nothing of this will happen with my amazing team over the years of students, postdocs, visiting scientists. So thank you. And if you have any questions, please let me know.